Hi everybody, and thanks for being here today. I'm Andrea Raffo, and in this talk I will summarize the results of our SHREC track on the retrieval and classification of protein surfaces equipped with physical and chemical properties. This contest is the result of a collaboration between Siena Remati and the Concept Lab at the Italian Institute of Technology, both located in Genoa. Behind this track there is an open problem in structural biology. When we think about proteins, we tend to consider them as static objects, that is, surfaces that do not change through time. Well, this is actually a misconception, and in fact, proteins do change shape, for example, because of their interactions with other molecules. Some shapes, though, are more probable than others, and the NMR technology can reveal which are more likely to appear. Given a set of protein surfaces, it is important to identify shapes that refer to the same protein at different times. For example, if we want to define a concept of similarity to compare proteins. I will now quickly go through the benchmark, starting from the dataset. For this track, we have created a dataset of about 5,000 surfaces. Each surface is saved as a triangle mesh, so basically an off file, which approximates the solvent-excluded surface. Each protein surface is computed by the freely available software NanoShaper, provided by the Italian Institute of Technology. The dataset is already split into a training set and a test set to help learning-based methods in the training process. For each surface, 3 scalar functions were made available. As a first property, we consider the electrostatic potential, probably the most familiar property of the 3 here considered. We computed it by using the Delphi finite differences-based solver, in a version integrated with NanoShaper. As a second property, we consider the hydrophobicity, which tells us which regions of a protein are more friendly with water and which not. Hydrophobicity is a piecewise constant function, resulting in the patchwork pattern shown in figure B. As a third and final property, we consider the presence of hydrogen bond donors and acceptors, which tells us where the protein is more keen to behave as an acid or a base. This protein results in the spot pattern that can be observed in figure C. The evaluation of methods proposed by the participants of this track is based on 2 ground truths. The first ground truth, which we will refer to as the PDB-based classification, is a 2-level classification of the dataset. In this ground truth, 2 surfaces are clustered together if they have the same PDB code in the protein data bank repository. Proteins having different PDB codes either refer to proteins having different amino acid sequence or to structures of the same protein acquire a different condition, for example, interacting with different molecules or having a limited number of point mutations. This first classification is the most strict, as it rewards methods that are particularly good in spotting minor differences between similar candidates. The second ground truth, which we will refer to as the blast-based classification, is a 4-level classification of the dataset. 2 surfaces can be either extremely similar, highly related, similar, or dissimilar, with respect to the concept of sequence similarity, which quantifies how similar amino acid sequences are. The sequence similarities were obtained by using a software called BLAST, popular in the bioinformatics domain. To ensure transitivity, a community-based algorithm was further used. We have considered a set of measures to evaluate the performance of methods to identify surfaces representing the same molecule at different times. More specifically, we have considered measures from the shape retrieval community, such as the precision recall curves, mean average precision, first and second tiers, normalized discounted cumulative gain, and average dynamic recall. Moreover, we have considered also classification measures coming from statistics, such as the true positive and negative rates, positive and negative predicted values, accuracy, and the Fisher score. I will now give an overview of the proposed methods, starting with an outline. So 8 groups registered to this track, 5 of which submitted the results. The participants were asked to consider 2 tasks. In task A, only the off files can be used, so basically only the geometry of the proteins can be exploited. In task B, the participants are invited to use both the off files and the corresponding physicochemical properties. The aim is to understand whether physicochemical properties can further improve retrieval and classification performance. Up to 3 runs per task were allowed. The variety of strategies was actually satisfying, as there were both traditional methods and more recent ones, based on machine learning. In this slide, you can have a look at the method proposed and the people that actually proposed them. So the first method estimates surface and volume-based shape descriptors, such as maximum and minimum curvatures and the area projection transform. Such descriptors are then combined into joint histograms and compared by using the Jeffrey divergence to obtain the similarity matrices. One of the 3 runs submitted by this participant considered a dimensionality reduction technique, namely the linear discriminant analysis, by directly applying it to the shape descriptors. The physicochemical properties were incorporated by partitioning the feature space into a set of regions, and then counting the number of vertices with features falling into each subregion. The second group proposed the use of 3-dimensional Zernike descriptors in a deep neural network framework. It takes 2 Zernike descriptors in the form of vectors of 121 features. Descriptors are then encoded. Encodings were used in 2 settings. In the first architecture, the encodings are further compared in terms of Euclidean distance, causing similarity, absolute difference, or dot product. Then the result of this process is passed to a fully connected network. The final output of this architecture is a probability that these 2 proteins belong to the same class or not. In the second architecture, encodings are directly provided as input to the fully connected network, thus ignoring the feature extractor layer. Being the method based on deep learning, the group split the training set we made available into a sub-training set and a validation set. In this slide, you can find some information regarding the training process and the validation accuracy, both for the case of only geometry and for the combined use of geometry and physicochemical properties. The third group adopted 2 distinct methods, the hybrid augmented point per signature, HAPS, and the histogram of process physicochemical properties of proteins, HP4-IDA. The first method, that can be seen in the figure on the left, robustly described 3D protein geometry represented with about 4,000 sample surface points. It involves concatenating 2 types of descriptors, an high-dimensional local augmented point-per feature descriptor with a 65-dimensional histogram of global distances descriptors. While the first type of descriptor captures the physical characteristics of a local 3D surface regions, the second type deals with the global configurations of the protein surface, by binding a set of normalized vectors between the centroid of a 3-dimensional protein and all the other points on its surface. The second method, which is schematized in the figure on the bottom, simply involves binding processed physicochemical properties and of protein surfaces into 1-dimensional histograms with various bin sizes, following 2 key stages. An initial exploratory data analysis, mainly consisting of outlier detection and removal for each property, and a subsequent combination of descriptive statistics such as mean, variance, correlation coefficients, and interquartile ranges. The strategy of the fourth group is based on a direct approach. Depending on the task, a collection of local and global features are calculated. For task A, example features include local curvature, volume, size of the minimum bounding box. For task B, other features are the mean intensity, standard deviation, mode value. Features are combined into a dissimilarity matrix where the unknown weights are estimated by using supervised learning. The last group consider graph neural networks, a deep learning strategy able of working with graphs rather than 2-dimensional images. The first architecture is called an EdgeConv. For each protein surface, the group first sampled 512 points on the surfaces of the input mesh, possibly considering trilinear interpolation on the physicochemical properties if a sample point is not a vertex on the original mesh. After this preprocessing step, the vertex features first goes through 4 layers of dynamic EdgeConv. Then the outputs of these 4 layers are concatenated and fed through another dynamic EdgeConv layer. The feature vectors are pooled using the concatenation of the outputs of a max pooling and a min pooling layers. This vector is passed through two MLP blocks with batch norm, leaky ReLU, and dropout layers to yield a 256-dimensional vector. Finally, the vector is passed through a fully connected layer to predict the protein domain. With the same data preprocessing procedure, the group also implements PointNet, a well-known graph-based learning method for three-dimensional data. In this network architecture, the vertex features first pass through two message passing modules. Each message passing module contains an MLP block that uses ReLU as an activation function. Finally, for each network, classification and retrieval on the test set are carried out by exploiting the L2 distances between representation vectors. We now present some of the results of the contest. Due to time constraints, I'm not able to give you a thorough analysis of all the measures, but I refer you to the paper for a more complete study. When considering a two-level classification, we can notice that all methods are more reliable in finding true negatives and negatives rather than true positive and positives. The overall accuracy of the approaches is quite high, being it always above 95%. However, a better analysis is given by the Fisher score, as classes are naturally unbalanced. The use of physical-chemical properties can add useful information, but it does not ensure improvements if not carefully handled. When considering a four-level classification, classification performance is generally improved, and this is maybe not surprising as the number of classes are reduced compared to the two-level classification. The two ground truths have actually much in common, and indeed, methods are good but not optimal. The highest mean average precision is 0,87 when only the geometry is considered, which increases to 0,96 when both geometry and physical-chemical properties are taken into account, being 1 the highest possible value. The strong heterogeneity in the class size has certainly influenced in a negative way the performance of the methods. Physical-chemical properties are a valuable resource, but they can also turn out being a problem if not correctly handled, and in this case, learning-based methods add problems dealing with them. In general, it is interesting to notice that deep learning does not guarantee better performance than more traditional methods, for example, because of data overfitting. To recap, compared to previous SHREC track contest on this topic, we have considered three physical-chemical properties. Moreover, we have provided the participants of a training set and a test set to make it possible for deep learning users to participate. Finally, we have proposed different ground truths on the basis of different problems coming from structural biology. The number of teams that register to the track and the actual number of participants confirm an interest of this community to problems arising on other fields of science, in this case from structural biology. Moreover, the methods were presented are actually representative of the number of paradigms nowadays available. We are about to apply to the replicability stamp initiative. The benchmark, as well as the runs submitted by the participants and the software used for the evaluation, will be made available on GitHub. Thanks for your time, and do not hesitate to contact me if you have any further questions.